So my work is studying how monogenic epilepsy might be amenable to gene replacement therapy. So our goal is to really use these more prevalently used gene therapy strategy, including AAV, adeno-associated virus approach, to deliver a gene in patients, and then to see whether this condition can be reversible. So we are starting from a preclinical aspects using mouse model, for example, and then to see if the condition in the mouse can be reversible by this gene therapy. So one of the projects that we are working in the lab. So first of all, maybe I want to introduce myself a little bit. So my name is Henry Lee. I'm a senior scientist in the FM Kirby Neurobiology Division in Boston Children's Hospital and affiliated with Harvard Medical School. And so what we are here doing is a gene therapy in the preclinical study, and focusing on a rare disorder called succinic semialdehyde dehydrogenase deficiency, SSADHD. So this is a rare disorder. Less than 500 patients has been documented, and it's a GABA metabolic disorder. So what it means is that in this disorder, the enzyme that's degrading GABA is a major inhibitory neurotransmitter in the brain. There's the recycling of this GABA molecule is impaired because of the absence of an enzyme called SSADH. So the patients basically cannot metabolize GABA in their brain. So that means they have GABA accumulation in the brain. And because this is also a second degree of degradation. So the pathway also push to a synthesis of a metabolic byproduct called gamma hydroxybutyrate. So basically patients have accumulation of GABA and GHB in their brain. And that makes, havoc in their brain because of the presence of these two neurochemicals. And the patients basically have intellectual disability, they have speech delay, and a lot of autistic features. And also, interestingly, as a scientific point of view, they have epilepsies. So it is a kind of fundamental question, why accumulation of a inhibitory neurotransmitter lead to overexcitation? So this is something at a scientific level we want to understand, but also this could be as a risk for gene therapy, because we in the gene therapy, what we want to achieve is to restore the enzyme. So you think about when you restore the enzyme, suddenly you have less GABA available in the brain, and less GABA means less inhibition. So the patients already have overexcitation. If you have less inhibition, what happened? It might be provoking more seizures. So this is not what we want to have. But at the same time, we want to see how gene therapy might be successful in this situation. So we use a mouse model to mimic this situation, basically putting a switch. Thanks to the genetic methodology that's available in the mouse, we can put a switch to switch off the gene. So we know precisely where the gene is in the mouse genome. So we can put a switch, what we call the lock-stop strategy. Basically, we put a stop codon into the intron of this relevant gene, and basically to stop the expression of the protein. So basically, at baseline, the animal has no enzyme expression, very much like the patient's situation. It is a autosomal recessive condition, so there's no enzyme activity can be detectable in the mouse. But because it is a switch, it's a lock-stop strategy, we can use a Cre-recombination to take out the stop codon. So that means by introducing the Cre-recombinase, then we can switch on the gene. All right. So by this method, then we can test a few things. One is that can we take out this stop codon? Can we restore the gene at certain time? Can we restore this gene at certain cell type by introducing the Cre at different time and different cell specific manner? Right. So that's exactly what we have tried in the mouse model. And the answer is yes. Actually, the animal, once they turn on the gene, they are receptive. They are at the baseline, they are symptomatic. They have seizures, pretty much like the patients. But when they are have the gene restored, they can grow back and the seizures suppress. So that means this seizure condition actually is reversible. And we look very deep into even their transcriptomic analysis, and just to ask the question, what happened at the molecular level? What happened to the transcripts? Is there any signs of, at a transcriptomic level, we can characterize the animal? And we also found very interesting results. There are many genes associated with seizure susceptibility, related to synapse formation, related to axon growth, axogenesis. Those genes are differentially expressed in a mouse model before they're rescued. But after they rescue with the Cre recombination, we see those genes are restored back to normal. So the implication behind that is that these genes are actually in a very different developmental trajectory, if you will. And when they are pathologically having a problem, these genes related to seizures are actually changing and indicating that it is related to seizures. But once the treatment is in place, many of these genes, if not all of them, are actually reversible back to a normal situation. So that means those genes are amenable, is responsive to gene therapy. So that's a very, a high level, very preclinical and theoretical level, because there's no patient have a switch for us to switch on the gene, right? So what we need to do is doing what everybody is doing for gene therapy space is now is doing AAV approach, delivering a gene using a vector. In this case, using a virus, delivering in the patient. So we are now working on that aspect and developing a virus which actually carrying a gene, and the gene will be delivered into the brain of the mouse, and then to see if we also can see the reversal of these phenotypes. And ongoing experiment suggested that the answer also is yes. Actually, the animal seems to be rescuable by this AAV approach. So that gives us hope about like how using gene therapy might be useful for some of these monogenic disorders and on monogenic epilepsies. I think the reasoning is behind that is because we know precisely where the genes are having a problem, and we can identify the target. And many of these genes are actually also fitting into the AAV capacity. So the virus has certain payload capacity, and if the gene is very large, then it might not fit into a single AAV molecule. So in those situations, there might be some other strategy that need to be used. But for our case, the protein is small enough to put into the virus. And we also think about the genes are not expressing universally inside the brain. And there are cells that are more, expressing this gene because of the GABAergic signaling, and the metabolic pathways of GABA is not universally the same inside the brain in different cell types. So that means the virus, wherever it goes, wherever it expresses, also need a certain regulatory mechanism for the physiologically relevant expression. So that's why in our vector design, we also think about that and using some of the promoter regulatory elements for the gene to regulate the expression of this relevant protein. So this is where we are, and we for the gene therapy workshop, we actually ask other experts in the field, especially a gene therapy space, Dr. Guangping Gao, Dr. Stephen Gray, and Dr. Manoj Patel. So they have been doing different stages of gene therapy, and that's our speaker in our workshop. And their presentations are also fascinating in terms of development of a vector or different regulatory strategies, or even beyond AAV. For example, Dr. Patel has talked about using an antisense oligo approach to increase the level of expression of other proteins. So these are the things that now we think that in the field of gene therapy for monogenic epilepsies might move towards, and there are relevant technology that it's maturing enough for us to use. And we're hoping that we know so much about the mechanisms behind epilepsies, and what is the next step using some of these therapeutic strategy to combine and join force to make that happen.